All right, well, thank you very much, Dr. Kloner, for those very kind words. Thank you for not calling me the grandmother of remote conditioning. I really appreciate that. And it's certainly my pleasure to be here tonight. So as Dr. Kloner mentioned, the classic cardioprotective paradigms of ischemic preconditioning and postconditioning involve application of the protective stimulus, that is, brief antecedent ischemia or stuttered reflow, directly to the heart. In contrast, remote ischemic conditioning, or RIC, involves application of the protective stimulus at a remote site. And the first evidence for this phenomenon was provided by our group, in which we applied remote ischemia in the circumflex bed and then applied prolonged coronary artery occlusion in the LAD bed in the anesthetized canine dog. And again, here we see the summary of these results. All animals underwent 1 hour of sustained LAD occlusion. This was either preceded by a no intervention period in the controls or 4 5-minute episodes of brief ischemia in the circumflex bed. And somewhat to our surprise, we found that infarct size was significantly reduced in the animals that received intracardiac remote ischemic preconditioning when compared with their time-matched controls. Among the first efforts to develop this concept, extrapolated from these observations of intracardiac remote conditioning to interorgan conditioning. And in one of the very first examples of that, we were able to show that transfer of coronary effluent from one buffer perfused heart to another could indeed transfer protection. In this case, we used isolated buffer perfused rabbit hearts. We had 2 groups of donor hearts. We had donor controls where effluent was collected after a no intervention period, or a donor preconditioned group where effluent was collected after 3 5-minute periods of global cardiac ischemia. That effluent was then applied to acceptor groups of hearts. And we found that, as expected, classic preconditioning in the donor hearts significantly reduced infarct size when compared with controls. Transfer of donor effluent from controls had absolutely no effect on infarct size, but again, somewhat remarkably, transfer of effluent from preconditioned hearts evoked a reduction in infarct size that was comparable to that seen with conventional ischemic preconditioning. But of course, the field really took off with evidence provided by Birnbaum and by Tarbanda and colleagues shown in this slide. Again, interorgan preconditioning, but in this case, as a preconditioning stimulus, applying skeletal muscle ischemia simply by inflating a blood pressure cuff on the leg of a pig. So in anesthetized pigs, again, all animals underwent 40 minutes of left anterior descending coronary artery occlusion. Animals were randomized to receive either 4 5-minute episodes of hind limb ischemia applied by a simple blood pressure cuff or a matched control period. And again, infarct size significantly reduced in the remote conditioned group versus control. And in the ensuing years, the concept has continued to develop very rapidly to the onset of small phase 2 clinical trials. And at the moment, there are at least 2 large phase 3 clinical trials in progress. But while the field has moved very rapidly in terms of clinical application, it has lagged behind somewhat in terms of characterizing the phenomenon of remote conditioning, and has lagged even further behind in developing an understanding of the mechanisms of this phenomenon. And you might say, well, so what? If it works, who cares if we know the mechanism? But I would argue this is critically important, and I think it's underscored by these facts. At the moment, we have approximately 25 published phase 2 clinical trials on remote conditioning, most of which were done in the arena of cardiac surgery. All involved application of 3 to 4 5-minute episodes of limb ischemia with the primary endpoint appropriately being infarct size reduction or its surrogate. But outcomes have been mixed. Just over half the studies showed a significant reduction of infarct size, but there are several studies that have reported that not only does a remote conditioning not work, it may actually be harmful. And then we have a couple of studies that were underpowered. And I would like to put to you that these disparate results may be a consequence of simple gaps in our understanding of the mechanisms of preconditioning. We don't know what we're doing in these patients. So what do we know about mechanisms? If we go back to the paradigm of classic pre- and post-conditioning, where all the action is taking place in the heart, the paradigm is reasonably well understood, although we're still a little fuzzy on some of the details. We know that the phenomenon is triggered probably by release of adenosine, bradykinin, opioids, and other compounds from the ischemic reperfused heart after the preconditioning or post-conditioning stimulus. These compounds then stimulate their respective G-protein coupled receptors and initiate one or more signal transduction pathways, colloquially known as the risk and safe pathways, involving extracellular related kinase, PI3 kinase AKT, JAK, and STAT3, and are thought to culminate on the end effector, most probably the mitochondria, and stabilization of the mitochondrial transition pore. But in contrast to this relatively simple paradigm in pre- and post-conditioning, the story does get a little more complicated in remote conditioning, and in particular, we have this added critical step, which is really the hallmark of remote conditioning, and that is communication. How does the trigger, presumably released from the limb in most of the studies, find its way to the heart? Now, certainly many studies have also looked at signaling with remote conditioning, but since time is short, I'm going to focus on the two leading hypotheses that try to explain this communication phase. I will tell you that in 1993, we made the suitably vague statement that the infarct-sparing effect of remote conditioning may be mediated by factors activated, produced, or transported throughout the heart during brief ischemia reperfusion. Well, now, in 2013, one of the leading theories is that this communication acts via a circulating humoral factor. Now, what might this factor be? Not surprisingly, given our historical interest in conventional pre- and post-conditioning, people have looked at adenosine, bradykinin, and opioids, and there's evidence for and against each and every single one of these agents as a potential trigger in remote conditioning. There is evidence from several studies obtained by HPLC fractionation of perfusate coming out of isolated buffer perfused hearts that what we are looking for is an as yet unidentified small, meaning less than 15 kilodalton, hydrophobic molecule. More recently, people have applied proteomics to this approach. And certainly there have been plenty of hits, plenty of candidates have emerged. Interestingly, the one uniform theme to come out of several of these proteomic studies is ApoA1, both from experimental studies and from a clinical screen of human plasma after brief limb ischemia. And this, of course, is also accompanied by several educated guesses. People have seen something go up in the plasma and have pursued this. And to show you just one example of what these studies typically look like, this is a recent paper published by Davidson, Yellen, and colleagues, where they were looking at stromal cell-derived factor 1, a chemokine that binds to its specific receptor CXCR4, and looking at the possible role of this in remote conditioning. And what they found in the in vivo rat model was indeed that plasma SDF1 alpha concentrations did go up after a remote ischemic stimulus when compared with control plasma. And then they went ahead in their model and administered an inhibitor to the CXCR4 receptors, which presumably attenuates this interaction. And what they found is that, again, as you would expect, significant reduction of infarct size with remote conditioning that was partially but not completely blunted by the inhibitor. And this is really what most of these studies look like. There seems to be something going on, but that evidence is really a little bit less than compelling that it's the entire story. Of course, the other questions that haven't been answered is, where are our factors or candidates coming from? I think everyone initially assumed it was the myocyte, but that certainly may not be true. It may be endothelial cells, it may even be fibroblasts. And how is this factor transported to the heart? Again, we initially thought probably in the blood or perfusate, but there's been an explosion of interest in transfer via exosomes or microparticles. And really none of these publications have seen the light of day. A lot of it comes from studies that have been done in brain remote conditioning, and this is kind of my insight in papers I've been reviewing over the last several weeks and months. Well, there is a second leading hypothesis, and that is that communication in remote conditioning occurs via neuronal pathways. And in this case, the hypothesis is that the source of the trigger occurs via activation of sensory neurons in the limb. This then stimulates afferent neuronal pathways leading to the central nervous system and potentially to the spine, and then transported, so-called, to the heart by activation of efferent neuronal pathways. This is the so-called remote preconditioning reflex that has really been championed by the Karin laboratories, and they in fact propose that every form of remote conditioning can be explained by this remote conditioning reflex. Now, I have a little trouble understanding how that may work in transfer of buffer from one heart to another, but again, they argue fairly vocally that they think it might be the case. I think a more moderate approach might be to say that communication in remote conditioning may well be model dependent. And in some models, the two hypotheses may be integrated. That is, we may have neurohumoral communication, again, possibly triggered by adenosine, and then affected by neural stimulation. And in submodels, this may in fact be redundant, that both mechanisms play a role. So in summary then, I read you our suitably vague quote from our 1993 paper. In 2013, I think we have several observations and associations that are absolutely intriguing, but to this point, I would say we have no real integrated, unifying hypothesis that explains the communication of this trigger to the heart to evoke remote protection. Thank you for your attention.